Hello, chapter 6, part 1. I would be lying if I told you this was going to be easy. Most students find that chapter 6 and 7 are the most difficult of the semester. Hopefully, we can get through it as best as possible. So we're going to start with cellular respiration. Next chapter will be photosynthesis. They're very similar processes. So when you learn one, you're kind of learning both because one is in one direction and one goes the opposite direction. So we're going to start with just some kind of introduction into this topic. So the first thing you need to understand is you need to understand. Whoop, thought there was another bullet that came down. The little saying oil rig, right? So for oil rig, what we're saying here is that oxidation is lost, reduction is gain. And so we can see here with A and B are both neutral. Now, A is going to lose an electron. So because we lost an electron, we have more protons. We have one more proton, so that gives us a positive charge. B gained the electron. So now we have an extra electron. So we have a negative charge. And generally, what happens is these two molecules will hang out together to negate the charge. And we talked about this with ionic bonding. Another way to look at this is it's a redox reaction. Redox is an abbreviation for reduction and oxidation because generally you cannot have one without the other. If one atom is losing, some other atom is gaining. All right. Electrons aren't just sitting in space. If they can find someone to hook up with. Okay. So here is that example of that ionic interaction. We know that sodium is a group 1 metal. Therefore, if it loses one of its electrons, it will have met octet. It will resemble a noble gas. So happily, it will give up its electron. Actually, let me do it this way. It will give up its electron to chlorine. Chlorine, if you look at chlorine, chlorine is a halogen. It is one column away from being a noble gas. So it just needs one electron to meet octet. So it will happily take that one electron. And so sodium, because it's being oxidized, and remember, oil oxidation is lost. So sodium is becoming oxidized. Rig, chlorine is gaining. Sorry, because, oh my goodness. I've got a piece of hair on my iPad. It's driving me nuts there. Sorry. All right. So because chlorine is gaining, right? We got the rig part. Reduction is gained. So it is being reduced. We are going to talk about all these redox reactions for the next 2 chapters. So you need to understand redox. So here is kind of the overall formula. The overall redox reaction in cellular respiration. So CR is cellular respiration. So we have, now you have to realize that hydrogen, if we look at hydrogen and we break it into its subatomic particles, it has one proton and it has one electron. No neutrons, remember, it doesn't have any neutrons. So when we gain or lose hydrogens, we are also gaining and losing an electron. So we can kind of use those together. So we have glucose. So when we look at glucose, glucose is carbon, hydrogen, and oxygen. When we look at oxygen, it's just oxygen. But notice when we go from here to here, what did we lose? We lost our hydrogen, which means we lost electrons. And which one's lost? Well, let's go through our saying. Oil rig, oxidation is lost. So notice we are oxidized. Here's our oxygen, right? Well, what happened to oxygen? Well, it picked up the hydrogen. And so it gained hydrogen, which equals electrons. So it gained electrons. And so when we talk about our oil rig, oxidation is lost, reduction is gain, boom, we're reduced. And in this process, we make ATP, which is what our goal is in this process. So basically, we're taking sugar and oxygen, We're going through a series of chemical reactions to give us carbon dioxide, water, and ATP. And we're going to learn exactly how we do this in the next few lessons. So we talk about potential energy. So again, we have a lot of potential energy here because of the position of this ball. Now, I like to use the example of, all right, you're at the top of this huge stairwell. Now, which one is going to hurt less? Slowly going down, bouncing, if you fall, you're bouncing down the stairs, or to just jump off and over the rail. Of course, if you just jump off and go over the rail, you have a ton. You are converting your potential energy to converting it to kinetic energy very fast, right? We can talk about the rate of gravity, 9.8 meters per second squared. It's gonna be fast. Or if we're just rolling and tumbling down, our position is the same. We haven't gained height. We're at the same height. We start off at the same position. But here we're slowly going downhill. We're going down fast. It's kind of like a bonfire. You throw a bunch of gasoline on it and boom, right? It's going to go up. But if you just let it slowly burn, we're going to get all the energy out of the chemical bonds in the wood. But which would you rather have, an explosion or slowly release it in that fire that you can enjoy over hours? And that's what we're doing with sugar. With sugar, we are going to try to slowly burn it and harness that energy very slowly versus letting it just catch on fire here. So one of the things you need to understand about this is the mitochondria. This is the organelle we'll be working in. And so with the mitochondria, we have two membranes. We have the inner membrane and the outer membrane. And then the space in between is called the intermembrane space. And then you've got the stuff inside, all this stuff inside, that's the matrix. And then we have these folds here. Those folds are the cristae. And those folds are in the inner membrane. And the reason why it's so folded or convoluted is because it gives it more surface area, because we're going to be doing a lot of chemical reactions inside that inner membrane. All right. Also inside there, so I have another picture here, which I like, because mitochondrial DNA. So chloroplasts, there's only two organelles that have, well, we could say three, right? The nucleus has one. We know about the DNA, our DNA in the nucleus. But we also have DNA in the chloroplast and in the mitochondria. And we talked about that with that endosymbiotic theory. So we do have mitochondria inside here in the matrix. We are also going to have their own ribosomes inside the matrix. So I like this picture because it's showing you that. So for the steps in cellular respiration, we're going to go through these in part 2. We're going to talk about glycolysis, intermediate step, Krebs cycle. The Krebs cycle is also called the citric acid cycle, ETC, electron transport chain, and chemiosmosis. All right, so we're going to talk about all 5 of these. So the next presentation is going to be much longer, right? So hopefully you have read up to this section. If you have any questions, please reach out to me. Do your guided reading if you find them helpful. If not, maybe at least go through and read the answer key. That might help you also if you don't want to hand do them all. All right, so just some suggestions on things that you could do there. All right, so that is it for this presentation. I will see you for the next one.